Hello everyone, my name is Xiaohua Lei from the Shenzhen Institute of Advanced Technology, Chinese Academy of Science. Thanks Dr. Chu for inviting me to attend this forum. I'm honored to be present our recent work on the study of mammalian embryonic development in space, and the title of my talk is Mammalian Reproduction and Embryonic Development under Space Conditions. Okay, let's see the development of the process in space bioscience research. They are divided into three development stages or around three major research content. The first is the study of biological system science. The macromolecular ways, the elegant zebrafish, fly are commonly used model. The research goal is uncovering the mechanism of space environment response. The second is the study of human health and space medicine, using tissues and organ, mammalian model, and human body to implore the effect of space environment on human health. The last one is to carry out cutting-edge research on human deep space exploration. The ultimate goal of its research is to implore that human beings can expand their territory beyond the Earth. As we know, changes in the gravitational field has significant effect on the physiology of living organisms, such as the development of bone, muscle, neurons, cardiovascular, immune systems will be affected under microgravity environment. However, microgravity-induced effective reproduction and embryo development had not been clearly defined. In the past few decades, numerous experiments on reproduction in space environments have been performed using sea ocean, amphibian, birds, fish, and fly. Most of previous results indicated that the process of the fertilization is not sensitive to the absence of gravity, and finding the fertilization rates were even comparable to that in parallel control at normal gravity. However, unlike the other Texas studies to date, mammalian early embryo from different species appears highly sensitive to the space environment. So far, the potential effects of wetness on the mammalian development are still unclear and controversial. Important works were the amphibian and the vertebrate development in space. In 1994, the xenopath egg was successfully fertilized, and some eggs can develop to the tadpoles with normal external in space. In 1998, Japanese macaque fish mated successfully in space, and young fish hatched during the 15 days of spaceflight. The obtained living offspring did not exhibit any changes in behavior or morphology and produce healthy second-generation fish when those fish go back on the ground. Previous study has attempted to examine the effect of spaceflight on mammalian mating and pregnancy by using unmanned biosatellites. After being sent into orbit to allow mating, the mature males and female rat failed to produce offspring, although post-flight examination showed that 2 out of 5 rats had signals of early pregnancy. Whether this is true for mammalian reproduction is still unclear due to the susceptibility of mammalian embryo to the cosmic environment during early development. There were several times attempts to study mammalian pre-implantation embryo development in space. In 1996, the 49 two-cell mouse embryo sent to the space on board the Columbia Space Shuttle non-developed. In 1996, among the 104 cell mouse embryo delivered to the ST-28 orbit module's platform, non-developed into blasts also. In addition, 601 cells borrowing embryo were delivered to the space. 547 were recovered, but no blasts were obtained from either group. Thus, we have still questioned whether gravity was required for mammalian embryogenesis. Technical difficulties and limited opportunities for spaceflight experiment in mammalian development encouraged some ground-based facility for simulation of microgravity to be used to study the effect of microgravity on fertilization and the pre-implantation development in mammalians. Kojima reported that the lump of fertilized egg that developed to morula and blastist is significantly reduced after 96 hours of culture under simulated microgravity. Wakayama reported that in vitro fertilized egg cultured under simulated microgravity conditions show slower development and fewer trophic cells in the blastist compared with normal gravity control, but no change in blastist polarization. Similarly, Wang reported that the effect of microgravity on cell growth at different embryonic stages is achieved via phosphorylation of SAPAC, and that SAPAC inhibitor can significantly reduce embryonic stress caused by microgravity. Nonetheless, those ground-based models cannot encapsulate the complex environment in space, and such as new technology need to be developed to enable such research in space. In this study, we want to answer two questions. One, whether mammalian pre-implantation embryo can develop from two-cell stage to blastist stage in space. Two, what is a potential mechanism that affects the development of mammalian pre-implantation embryo in space? We developed an automated mini incubator to enable mouse early development in space that was equipped with programmable controls for temperature, automatic microgravity, and the fixation of samples for cultivating mouse embryo. We cultured two-cell embryo for 64 hours under conventional culture in the microgel carbon dioxide incubator or sealed culture in developed automatic mini incubator conditions and compare the experimental outcomes. The automatic mini incubator allows the pre-implantation mouse embryo to develop normally under the sealed cultural conditions, and is therefore suitable for subsequent experiment in spaceflight. To assess mouse pre-implantation embryonic development during spaceflight, 3,400 morphologically normal mouse embryos at the two-cell stage were placed into 4 image culture chambers and 6 culture fixation units. This is image culture chamber. Time-lapse imaging demonstrated that two-cell embryo culture in space underwent key stage of mammalian pre-implantation development, including cleavage, compaction of plasminoles, cavitations, and the formation of expanded blasts. We further assessed the morphological and the rate of development of the embryo in fixation culture unit, returned from the space. Of the 1,500 embryo loading, 1,118 cells were recovered and consisted of compacted morula and expanded blasts. Among them, the majority developed into morula or blasts, but the ratio of the blast stage was significantly lower than that of the conventional control and the sealed control group. Thus, two-cell mouse embryo can develop into blasts in space, but the ratio of embryos developed to the blast stage is greatly compromised. After seeing the rate of development, we examined the quality of the blasts developed in space and found that the total number of cells in the blasts developed in decreased significantly. We also performed immunostaining of CDX2 or trifactor marker and OCT4, an inner cell mass marker in blast stage embryo. Interestingly, there was a significant increase in the percentage of OCT4 positive cells in space embryo. In contrast, the percentage of CDX2 positive cells was reduced in space embryo. In addition, the percentage of CDX2 positive and OCT4 positive cells was significantly higher in the SS embryos than in the SC and CC embryos. To investigate the potential underlying mechanisms, we first detected whether DNA damage occurs in embryos developed in space environment. We examined the formation of gamma H2A6 and 15-3BP1 forces by using their two biomarkers for double-strand break of DNA repair, and the XCC1, a marker for efficient repair of DNA single-strand break. Quantification of fluorescence intensity confirmed a significant increase in gamma H2AX 15-3BP1 and XRCC1 expression in SS blasts in compared with the ground culture embryos. As we know, epigenetic regulation, such as DNA methylation, play a vital role in early mammalian embryonic development. The developmental defect in space embryo led us to ask whether they are linked to the change in DNA methylation during pre-implantation development. We perform genome-wide DNA methylation profiling of individuals' blasts using bisulfite sequence. The results showed that the percentage of global cytosine guanine methylation in space blasts was significantly lower than that in ground embryos. We further analyzed DMRs, allowing us to identify 12,517 DMR between the space and the ground conditions, and 11,498 DMR between the SS and SC conditions. There were higher DMR with the lower level global methylation in space blasts. Functional enrichment analysis indicated that the low methylation inclusive DMR in the SS blasts were related merely to histone modification, response to radiation, regulation of chromosome organization, cytoskeletal organization, and others, while high methylation inclusive DMR for CC blasts were associated with embryonic development, regulation of RNA metabolic process, and the regulation of intracellular protein transport. Those results suggest that, in response to the environmental factor in space, the genes responsible for histone modification and the radiation are hypomethylated, leading to their activation and the subsequent repair of DNA damage. The mouse pre-implantation embryo in space are exposed to two main environmental factors, radiation and microgravity. To ask what factors might have caused their development, genetic and epigenetic abnormality in the embryos, we import ground-based low-dose radiation to mimic the environmental inside the SJ-10 satellite. We found that most two-cell embryos exposed to 0.1 Gray for 64 hours can develop to the blast stage. However, when the dose was increased to 0.5 microgray, only 58.4% of the embryos reached the blast stage, and the percentage decreased to 45.6% when the dose was increased to 2 microgray. Consistently with the development defect, we detected the fluorescence forces positive for gamma H286X in embryos exposed to 0.5 microgray or 2 microgray, with the forces more intense with 2 micrograys exposed. We also find that there was a statistically significant decrease in genomic with-right DNA methylation in embryo with 2 microgray exposed, and with a higher percentage of hypomethylation DMRs when compared with the control, but not with the 0.1 and 0.5 microgray exposed. Additions, genes with different methylation were enriched in cellular process, such as response to radiation, response to stress, and cytoskeletal organization, regulation of histone modification, regulation of histone acylation, and the regulation of protein ubiquitination. To examine the long-term consequence of radiation, we determine whether the embryo with radiation exposed can develop to full-term mice. We transfer the exposure were structurally lower than that from embryo without radiation exposed. We further ask whether microgravity can also cause the defect in pre-implantation embryos. We cultured two-cell mouse embryo in a rotary cell culture system. we found that most two-cell embryo exposed to SMG for 64 hours could develop to the blastocyst without statistically significant difference when compared with normal gravity. Furthermore, we failed to observe significant difference in DNA damage and the global DNA methylation level between control blastocysts and those developed with SMG. So, take-home messaging. 1. We overcame technical difficulties and developed an automatic mini incubator that enabled to examine development of two-cell during spaceflight in real time. 2. We show unequivocally that pre-implantation mouse embryo can develop in space, although the rate of blast formation and the blast quality are compromised. 3. The cells in the embryo contain serious DNA damage. Additionally, the genome of the blastocyst developed in space is globally hypermethylated with a unique set of different methylation regions. 4. The development epigenetic abnormals can be largely mimicked by the use of ground-based low-dose radiation, but not simulated microgravity. This work has been published in National Science Review as a cover story. In addition, our work has been commented by Professor Magdalena Zienig-Grose from California Institute of Technology. The development of mammalian pre-implantation embryo is only a small step, but there is still a lack of knowledge about the mammalian reproduction and development in the space environment, and many unknown scientific problems and mysteries need to be further explored. For example, can space blastocysts produce offspring after transfer? Therefore, it is necessary to make sure of spaceflight platforms such as space station and the scientific experiments satellite, assisted by ground-based simulation of microgravity, radiation, and their combined effects, to further systematically carry out study on reproductive function and development. It focused on the effect of space environmental factors on follicular development and oocyte maturation, spermatogenesis and maturation, fertilization and pre-implantation development, and post-implantation and fetus development. Finally, I would like to thank the grant from the CIS, NSFC, MSTC, CMS for support in this project. We thank all the experts for guiding in science and technology. I also would like to thank all the authors for help in their study. That's all. Thank you for your attention.